And let's continue with the next presentation. Let's go with a very interesting custom microscopy talk. Our next speaker is Thierry Emeraud. He's Business Development Director at Lambda-X. And yeah, I'm very curious about your presentation to see what's your developments for bioimaging. So the floor is yours.
Okay, thank you very much, Antonio. Can you see my presentation full screen?
Yeah, we can.
Great. So thank you for the opportunity to introduce our contribution to this meeting. And I'm Thierry Emeraud. I'm a Business Development Director at Lambda-X. And as a short introduction, I think it's interesting. Maybe we'll have the possibility to look at this bioimaging more in an engineering perspective than a fundamental science perspective, as was presented by the former speakers. So outline of my talk, I will shortly introduce who is Lambda-X, who we are and what we do, just in one slide. Then I will go through three different use cases that we have contributed to in bioimaging, and then we will go through the traditional epic questions. So Lambda-X, we are an engineering and providing engineering services and OEM manufacturing services for custom optical systems. We serve different markets, as you can see here. However, today we focus more on the medtech and the life science market. We are close to 50 people, and we have contributed to 300 product development projects so far. And I would say our key contribution can be summarized this way: explore widely, I would say, different optical concepts and also engineering solution at whatever step of a product development stage, and co-create with our customer and globally de-risk product development projects at our customers. So now going through the different use cases. So the first one is a custom microscope that we contributed to make robust and reliable, and which is used for enzymatic quality control and process control. So our customer has developed biopolymer microstructure deposited on silicon plates, which they interacting with some materials which is used in different processes using enzymes. And could be in the food industry, could be in the detergent industry. And what we do and what they do in the end is, as you can see on the top right, is to analyze the shape and the volume of the modified biopolymer structures after their interaction with the material to test or the process to test. And from then on, they get a qualitative information on how big is the enzymatic activity. So you can see some pictures here showing the microscope, and what we have been doing is to make an initial design which was quite unstable to make it robust, reliable with no instrument-to-instrument variation. We had a design to cost contribution to by making and developing a custom autofocus system. And today we are a partner for this company to manufacture in quantity 10 to 50 units per year. The second use case is about mid-infrared spectrometry and imaging on biopsies. And here we work for a company which is called Admir, which is a spin-off from Cevatec in Grenoble in France. So what is the context of the application? It is basically oncology. There are more and more tests to be performed on a growing number of patients. However, we face some shortage in anatomopathologists, and which results in a very, very small fraction of the biopsies which are currently read. So the idea of Admir was to propose an automated fast infrared imaging, and you could see it as bringing a well-known lab technology, which is infrared fast Fourier transform infrared spectrometry into a very fast device by combining three key technology bricks. First is the high brilliance infrared lasers, a bolometric matrix as a sensor, and bringing some artificial intelligence for image analysis. We can see some results here. So you can see on the top left the traditional staining image you will see from a traditional biopsy, and you see that with the type of technology you can get similar images. However, on top of this, you can make some tissue classification using this artificial intelligence software contribution. And what have we, and you can see also on the bottom left, the demonstrator, which has been built by Admir and already tested in some marketplaces. So what we have done here, we have made the original concept, and we have designed and manufactured a custom illumination box for that microscope, which is made of 8 multiplexed quantum cascade laser sources operating in the range 5 to 11 micrometer. And the challenge for us was to deliver an homogeneous illumination, and particularly managing speckles from those Korean sources, and reach better than 10% homogeneity over several square millimeter area. And on the other end, to reach a high enough signal over noise ratio to enable the fast measurement. As I mentioned, the target is to perform the images in less than a minute. So the third use case I'd like to introduce is different. It's about cell therapy, and especially one key step in this cell therapy process, which is cell expansion. After the cells have been transfected, you need to monitor how they develop and how they are transforming after the transfection over time. So here we talk about another kind of microscope, which is a digital holographic microscope, which is performing the online monitoring of the cell population all throughout the process and online. That means you can really monitor the morphology of the cell by a label-free 3D live imaging. So as you can see here, when you start, you get some kind of uniform cell shape or morphology, but as time goes, after the transfection, the cell try to change, and actually the technology enables to discriminate between an infected cell, a dead cell, and a viable cell. And in the end, you can make a clear cell population diagnostic, which tells the pathologist or to decide what you should do with the material in the bioreactor. So here, what Lambda-X has done is the industrialization of the microscope to reach full GMP and UL compliancy, because the major customer was in the US. And today we are also an OEM manufacturing partner to the Ovidio company. And we have built over 200 units over the past 3 years. So that was the 3 examples I'd like to highlight how engineering can contribute to bioimaging applications. And to end up my talk, we could go shortly through the EPIC questions. So what can we do for you? We would like to be your partner to innovate with optics for your products, for your whatever device. And we can do that as Emeraud. We like to elaborate for new product concept or to de-risk product development as a whole. And what could you do for us? We are eager always to work on new bioimaging application with optics in a very general perspective. And we could contribute to test or to integrate innovative components for such application. And well, maybe those will trigger some questions from the audience, but I would like to thank you for your attention, and I would be glad to answer whatever question you might have.
Thank you very much, Thierry, for showing some of your capabilities and also proposals to collaborate with other companies. Any question in the room? Yeah, Alexander from Toptica.
Yes, just if I noted correctly, that you use 8 multiplex quantum cascade lasers to achieve the performance for the device. Is that correct?
Sorry, could you repeat the question? I didn't get it at the beginning.
I made a note that you use 8 multiplexed quantum cascade lasers, right? So my obvious question is if this should be potentially better to have maybe one source which covers at least this wider range that should make the setup potentially better. And maybe in other words, what other problems are you facing with the such a big amount of quantum cascade lasers?
Well, actually, the customer decided to do it this way to get fast, because they selected the 8 different quantum cascade lasers because they were emitting at the wavelength of interest, and depending on the application and the type of molecules you want to probe, they will change those sources. But they say that, well, in a whole spectrum, or in a distributed spectrum, there are wavelengths we are more of interest, because you will probe the appropriate vibrations regarding to some molecules. So first they select which laser source they want according to the wavelengths, and at the end it makes the device easier to industrialize in this way as compared to another type of source.
Thank you.
Thank you, Alexander. We had again the same question, but you were first now. Another question for Thierry. I also have a curiosity regarding your solution for measuring the particles or distinguish which particles were cells were infected and no. Which are the advantages over other techniques like the nanoparticle tracking analysis, other kind of techniques available in the market? I guess you made a solution quite customized for the application itself, right?
Yes, actually, so this is a digital holography, so it has a lot of advantage. First, the best advantage they have is that they can monitor online. That means they don't need to take any biomaterial from the bioreactor. So it is made in a way that the biomaterial is circulated in the focus of the microscope. So it's a really online technology. There is no interaction with the biomedium. So this is the first one. And then this digital holography technique is, well, it's label-free. Of course, that's important. It doesn't need to involve any marker. And it is performing a volumetric analysis, which is quite interesting. And well, so far, that's, and also it's, I think it's a self-calibrated technique. So, well, according to our customer, again, we develop product for customers. So the ultimate advantage they could bring on their competing technology shall better be answered by them. Well, that's the way I can answer to your question.
Mm-hmm. Yeah, good. Thank you. Elizabeth from Hubner.
Thanks, Thierry. Very interesting to hear all your applications. I had a question, the digital holography application there. Do you use lasers for that?
Yes, indeed. Indeed, they use a laser source. I cannot disclose all the detail, but it's well, I don't which type of laser diode or specific source. I cannot tell exactly now. Maybe I could.
Sure. No, I understand. No problem. Yeah. Thanks.
Perhaps in a private conversation later on. Good. So thank you, Thierry. Really nice presentation. And I tell you that I was really amazed when we talked first time and you showed me all the capabilities of Lambda-X. So feel free to explore the website and see what can they do for you. And let's move to the next presentation. We are talking about